There is 11 million people dying on sepsis per year, and it's increasing. 80% of this, imagine, could actually be maybe not happen if there is an early diagnostics. You always realize that you are always late. By the time that you apply the therapy, it's too late. The aim of NIVD is to create something simple that does not even require any little machine.
I really enjoyed this conversation with Dr. Sonia Trigueros. She's the founder of NIVD, and she's working on nanotechnology that can detect diseases way earlier than anything we have today. The way she explained how molecules shift at that scale was fascinating. She even offered me a site visit, which I'm seriously considering. Sonia has this great blend of academic depth and practical curiosity. We talked about her move from Oxford into building a company, the science behind her early detection sensors, and how this kind of technology could change healthcare in a truly meaningful way. Just a little bit, about your background and into academia, your time at Oxford. You can go back as early as you want.
And then... Oh my God, okay. I'm a biologist for Barcelona University. So I started in Barcelona. Then I did my master's in Biotech in Barcelona University as well. And then I did my PhD in molecular genetics in Barcelona as well. So this is the beginning of your scientific life, okay?
Okay.
Then by the time you obtain your doctorate degree, okay, and I really wanted to do science, so I decided to carry on with the academic life, where in my head, it was more like doing research and discovering things. I mean, I like very much innovation, as you will see over the conversations. I felt extremely comfortable. And when you finish your PhD, you start a series of phases called postdoc after your doctorate, which are a little bit risk because you change a lot of different countries, you change places until you get enough publications, enough data, enough money on projects to become a full professor, okay? It's a very tough situation, but it's like the life of after a PhD, which is crazy. And so my first postdoc was at Harvard University, and my second postdoc was in Oxford, and everything started there. One of the things I've been involved in biology has always been working on how DNA is packed into cells, why you have 2 meters of DNA per cell, okay, with only 5% of that DNA give you genetic component. That's totally true. Every single cell of your body, you have 2 meters, right? But only 5% of these 2 meters have a genetic information. If you have 2 meters, it's because there is a biological reason. What is this biological reason? Why we have 2 meters, not 1? That's what more or less bring me into science, the curiosity of why. So I developed my career through my PhD and my first postdoc on discovering when this DNA is not well packed, because 2 meters in a very tiny space have to be very well packed, not noted, not broke. What happens when this fails? So what proteins regulate everything, and what happens when these proteins don't work properly? Basically, my first postdoc at Harvard University, I was very much studying the relationship between these proteins and early aging diseases like Bloom syndrome, Werner syndrome, are syndromes that people have when they turn 30, 40, they start to look like 80, and they have tumors, they have Alzheimer's, they age extremely quickly. So the question is, why proteins that control the DNA have this powerful tool to turn your life into these diseases, right? So I become kind of a molecular detective in that moment. I'm saying always, why all this? I love it. Really like it very much. With that, we find out as well that when these proteins are overexpressed, you have a lot. That's one of the first indications that this cell is growing really fast, right? So if you have a protein that has to grow faster, this DNA have to go from 2 to 4 meters, have to duplicate to generate the daughter cell. So you need a lot of these proteins. And that's the definition of a tumor cell, okay? So a lot of these proteins that I work from my PhD until now are the targets of chemotherapy drugs. So by blocking these proteins, you block the exponential growth of tumor cells. This is a little bit more or less the background where I'm coming from. So I study from like the growth of tumor cells as well, bacteria cells, virus. So I become very interdisciplinary from very early stage in my career. I like very much the intersection between infectious disease and diseases that at this moment, we don't think there is any correlation, but there is a lot. And then I moved to Oxford. I thought for a postdoc, Originally, I was supposed to be there for 2 years, but I was 17 years, so I like it there.
Dude, yeah, I hope you liked it for the
I like it
17
very
years.
much. I mean, it was a place where not only I developed my full potential as a scientist, but I have the opportunity to become a founder and then director of the Institute of Nanomed. I work as an advisor in the Said Business School at the Biotech School, always bringing the science to stakeholders, right? And I was very much involved on if my science have to be something useful, who makes the decisions, how I can move that. During that period of time, I developed a technology that my idea was to develop a technology using nano, and it was a technology that helped me to carry drugs to a specific tumor cell rather than the whole your body. As you know now, when you go to chemotherapy or when you take a paracetamol, any medicines that you take now, only 3% of the medicine reach the part that it needs to target, because most of it is destroyed by the saliva, by the stomach, it has to go to the bloodstream, and then it reaches the cell, has to enter into the cell. And then at that point, I said, well, I start to play with nanotechnology. I mean, it was very much physics rather than biology. So within Oxford, I have the opportunity to move to the physics department. And that was very weird for me because, I mean, from biology to physics, we don't understand each other. They were very nice people. I mean, I learned a lot. I developed new technologies using nano, and this invention works. And so we have a very interesting technology to actually bring chemotherapy drugs and any other thing to specific cells. However, when I was doing the experiments, I realized that when I started the therapy was the problem. So when I started, in which moment I have enough tumor cells to start a therapy? That was in the lab environment. And then I started thinking, when is the people starting therapy? How the tumor is diagnosed, right? And when you start thinking like this and you see how the diagnosis system works at this moment, you always realize that you are always late. By the time that you apply the therapy, it's too late. It's always the disease is always there. You have symptoms because the diagnostics comes from you have symptoms. That's why you go to the doctor, right? Yeah. Okay. So when you are in the lab, you think, this therapy works well, but if I could start earlier, it will work better because there are less tumor cells. How early is early? So I decided to develop some sensors using nanotechnology to help me identify proteins or molecules, any molecules, not only proteins, that was telling me at the very small level, hey, something has already started. You cannot see it yet, but it's starting yet. It will happen in a few hours or in a few years, right? So I developed these sensors, and one of the things I thought, that is interesting. I was using a complicated nano moment. Yes. No, it's not. And then I realized that it's not just one protein or one molecule. There are hormones. There are other parts of the cells that comes from my Harvard previous work that could inform me that something is going on in your body. So the technology initially I was developed for 7 different types of molecules. So to detect DNA, hormones, sugar, lipids, proteins, anything that is wrong in your body that shouldn't be there in order to tell me, hey, Sonia, this has started applied therapy. So I did it and it was extremely useful, really useful. And I thought, wow, this can actually help the current diagnostics because diagnostics, we are always late. It is very expensive. Okay. Yeah. And the doctors are doing their best with the tools they have. But what about if I could actually take this, make more than 7, make like a library of different sensors to detect as many molecules possible in different diseases? Can this help to the doctors? Can I do something that is so easy to do that the doctors can do it in surgery, can do it in this, like a real point of care? And this is NIVD. This is NIVD. NIVD has produced a library of sensors, to detect diseases before you have symptoms. It's extremely fast, extremely cheap. We use nanotechnology for overcome problems of current technologies, and we work with quantum technology, so it's really fast. When we actually saw that this is useful, that we can scale up, then NIVD start growing more and more and more. But NIVD is the vehicle that, with a group of people, obviously, I haven't done NIVD alone, not alone, absolutely not. But I thought this has to see the light. I want the doctors having this technology in hospitals to detect things before you have any symptoms, during a surgery, or at home or in any point of care or in places where there is no hospitals. And that's the story of NIVD right there.
That's
Yeah.
exciting. That's exciting. So you started with looking at delivery, drug delivery, more precise delivery, and then that transitioned into
Yeah,
detection.
I was more in the delivery to stop the tumor cells or bacteria cells to grow when I realized that we have incredible therapies right now in the market. We have immunotherapies, we have all new technologies, immunomodulators, we have incredible technologies, but they fail or they are not optimal because we start always extremely late. But we are working right now with sepsis, okay? By the time the sepsis is diagnosed to you, they give you antibiotic. But what about if we can understand the sepsis has started before you have the fever? Then we can actually detect this. We can detect, is this it's a bacteria, it's a virus? In which moment are you? It's already the immune system not working properly because that's the sepsis. Sepsis is not an infection. It's a misregulation of your immune system. And we do have current therapies that we cannot work properly with them because we don't know this. So we are early because we interrogate the system extremely early. And we are asking questions, okay, with our technology, and we find things before anybody else because there's no other technology. They're super cool.
It's super cool. Yeah, and on that note, I want to dive a little deeper into like what exactly nanotechnology for those who don't really that term and just how you would explain that.
Yeah, so nano is a size. It's a size, okay? Now, technology is, or nanoscience, we work on materials at very, very, very low size, okay? It's 1 billion part of a meter. However, there are two important things at the field of nano that it really catch my curiosity. The first was the size equals size of parts of your cells. So it's like you are touching things, right? Like the cells, they're the same size of biological molecules. That's the first important thing. But second, okay, and that's why I dedicate my life into nanomed, is when you work with inorganic material at the nano level, the material, when it's so small, they have completely new properties. But properties at the magnetic properties, semiconductors are all different, photonics, they change completely because the material at the nano level is ruled by completely different forces. Some of the nanoparticles don't even feel gravity. Can you imagine? Because exactly. It's what years ago they were saying at the atom, when you put 2 atoms, 3 atoms, 4 atoms together, still in a moment, the material is still not like at the atom level, is not like a big bank of level. It has very interactions between the parts of the atom. That's the quantic behavior of the material. So that's where we work at that level. And in that way, you have material that is faster in terms of semiconductor or has different photonic properties. And then you tune this, okay? You can tune these properties when you understand the material, which took ages. And you tune these properties to do what you want them to do. And that's what NIVD did. We create some material that it makes a change on one of the properties. And for that change, we figure out, okay, that molecule is present. Okay, so nano is telling us this molecule is there or is not there.
So, and that's by how the nano reacts once it's in the body.
You make it, but we are in vitro, right? We don't have to put anything in the body. We develop the technology with a team to make it able to use it in saliva, in blood, in urine samples, in liquid biopsies, in any biological. Take it out. It's so simple and fast. Okay?
Yeah.
Take it out, you mix it, and quickly, because it's at the quantum level, you say there is this molecule or there is not. And by knowing, you start looking at the scenario of this person. Okay, that person is starting to having a period of sepsis, is not, it's very early. Okay, so we can apply this antibiotic rather than broad spectrum, will be much more effective. So we're trying to help diagnostics and we're trying to help the efficiency of the current therapies that we have and not to be overdosed later on some medicines because it's too late. And we create that by using nanomaterials that we tune to obtain a different property, and the change of the property is what is our answer. That's more or less it.
More or less, yeah. Simple, very simple. I want to see it. I want to go to the lab and like
Oh, you
see
should come.
what
Yeah,
is it.
please, please. Absolutely. I mean, everybody that comes to Barcelona to see us, we do a demo test. Yeah, and you could feel it how
And it seems
quickly
like that's pretty scalable.
it is. That's the so that was the important part that we developed over the last year. In order to, I mean, if you build a company, one of the most important things in biotech is to make sure that it's scalable, that you can produce many of
Yeah.
them, and that has been the effort over the last year, and we can actually make a lot of them, so it's cheap, we can scale up. There are two challenges, obviously. Well, no, it's all about a challenge but there are 15 years of research behind this, right?
Yeah.
And
So you went to school
we
for a long time.
and it's not only on nano, but all the knowledge on how bacteria and the cells work, and all the molecules. So it's really fun because in your day you have physics, chemistry, biology, medicine all together. So sometimes you feel you don't know nothing about anything. And hence that's important of my team. The team is essential, not only the scientists. We built as well a team for the commercialization because that's really important. I mean, it's impossible to do these things alone. So I don't
Yeah.
want to get any granted for this. Absolutely not. And the team is great. And now we're working to take this because we are already in hospitals. We can see how useful it is. My aim is to see this in with the hospitals. That will be my aim. Totally. It will
Yeah.
be make me very happy that I can help doctors to make a more accurate diagnosis. Yeah.
Yeah. And you mentioned that the team, I feel like always is such an important part. So like, how did
Yeah.
that come together? How did you find your people?
Many ways. I mean, uh, most of the times, believe it or not, it has been for them having conversations with people, with friends. I mean, we have like a nice network of people, and basically conversation with people that are completely from the business side, completely different side of me, but they're very interested in understanding the biology, because at the end of the day, we all go through this process, right? We all have an infection, or we always know somebody with a tumor, and we start talking and talking and talking, and I always say that they get infected with NIVD and then they've been together. So yeah, it's beautiful what we're doing, and I think it's useful. So people are putting the effort and the time to help us to grow. And so we have advisors, we have people already employees, of course, and we welcome every kind of help, to be honest, because I think that is something that we need to
Yeah.
be hospitals. So no, finding the people, I mean, then when you have like you make your own, you first the first round of funding, you can start building the employees. The employees that we're looking for has to be, they're very special. I mean, the ones I have, I love them all. I mean, they are really my family now, but they have to be people that are not scared of innovation, because most of
Yeah.
the time things like what's going on, like I don't know. I mean, innovation is overrated, to be honest. And then they have to be people open-minded to learn from physics, medicine, commercialization, structure of the company, because we all one pack. You cannot have a technology in the hospital, so you don't have a very good commercial team. My role right now is to make sure that everything is aligned, right? So we have to align commercialization needs, science, legal, all that, building the company. Yeah, having fun.
Yeah. No, fun is important too.
Yeah,
Yeah.
well, I won't do it if it was not funny. No, I won't do it. No.
Yeah. It's too hard. It's too hard to not have some fun too. And talking to others who making that transition from academia
Uh-huh.
to the business world, what were maybe some of the lessons that you've learned or just challenges, any challenges associated with leaving that environment?
I don't feel that I left academia because I'm an associate professor at the University of Barcelona. I left obviously because I want to build a startup. It was very natural for me. It's
Hmm.
not like I thought about it. Okay, there is a risk because you are like comfortable because you finally get to your position where you know that you have fight so much. But all my life since the beginning, I didn't think that much on this. It was not that important to me to become the best professor in the university. I want to do something useful. When you build something, you want this to be out. It was natural for me to make this transition. If I want to make this happen, I have to put myself into it. And so I tried. I thought, well, if I don't like it or I feel like overwhelmed from the unknown, that will bring to me, maybe I can quit or something. I mean, why not?
Yeah.
But I mean, I have a very good trial in terms in Oxford University, because this is my second patent. So I already have one patent first. I was very much exposed to commercialization. So I didn't know nothing. I learned everything with NIVD. It didn't felt that, oh, I stopped this and I'm starting this. No, it's like a continuation. It's like a book, right? I mean, a lot of people say, yeah, no, I changed. For me, it was like, okay, this has to go to the hospitals. I have to do it.
Have to get it out there.
Yeah.
Yeah. Yeah, that's inspiring. I was going to ask you for advice for anybody in academia that wants to make the transition, but it feels like you answered that. Just don't think about it. Is that?
Well, it's not that don't think about it. I mean, when you're in academia, that's something that I didn't like it very much, but I didn't know. Some people see science in the commercials part, right? Or in the companies, they see that like we do a different type of science. So in academia, you do more like a research, and the other one you more implemented a product. But at the same time, it's all about science, right? It's just different part. So you don't know which part you will enjoy more. You don't know if you are, because when you finish your PhD, you don't know nothing. Sorry, but that's the reality. It's during your postdoc life when you start learning what academia is, where you see yourself doing in the projection in the future. Okay, you have to find your own path. And if you feel that you want to try it, why not try it? I mean, there is no black and white. You can always come back.
That's a change. Yeah, you can
I
try
mean,
it.
advice is try it, but not alone. I always say, have a good
Yeah.
team. Yep.
Yeah, I feel like that's a key piece too. You can try it. You can always go back, but also choose the right people to
Yeah.
do it
Oh
with.
my God, it's so important. And always open-minded, because they are very different type of worlds, right? I mean, the way for everything from payrolls to the way that is daily managing is completely different world. And I think that one day, I really hope that they merge a little bit, because I see parts very strategic and very productive in the commercial side, right? And then the scientific way or the thinking way from academia, it's actually very good. So I think that hopefully NIVD is a little bit of a mix of both. Who knows?
Yeah.
I hope.
No, I feel like it could be with your background. Like those two things can merge. I feel like there's space for both.
Oh yeah, there is. I mean, in science, look, science is a space that everybody wants. You just need to try it, right?
Yeah.
People don't be scared of trying new things. Well, I mean, if you are scared of trying new things, don't try it.
Yeah. Well, it's like you said, I feel like there's so many amazing products or ideas that are being created in lab spaces in academia, but to get them out there into the
Please,
world,
no, no,
into real
it's a
world
competitive.
application.
Exactly. I mean, I have seen incredible ideas, incredible. Some of them they try. I mean, it's very difficult. I mean, it's very complicated, not easy at all. But go for it. Try it.
Yeah, go for it. Go for it.
Yeah.
And on that, where are things at now?
Well, we are now, I mean, we are still very young startup. Where we are is creating still the very good base. We are developing a lot of sensors. We're proving that we can scale up. We are on commercialization sites. So we are growing very slow, but in a very good base. Okay. And then exponential.
Okay.
We are in the first product, with the first disease. And then as soon as we have the first disease going on in the commercialization, we can start with the next one.
Okay.
And
What
then
is that first disease?
it's sepsis. We are in
Sepsis.
sepsis, right?
That is sepsis.
I mean,
Oh, that's a big one.
sepsis is a very interesting disease. I mean, we know now that is actually there is 11 million people dying on sepsis per year, and it's increasing. 80% of this, imagine, could actually be maybe not happen if there is an early diagnostics. So I'm sorry, but there is a huge need for early diagnostics in sepsis. I mean, it goes really fast. It's started by a bacteria or infection, and then goes, because it could be a virus as well or a parasite, and it goes in days. If you have like one organ only damage, you have a sepsis, then you can sever sepsis because you have more than one organ. And then when sepsis is spread, you have more than two organs, and you will know that it's called multi-organ failure. It happens not only in elder people, it happens in young kids, it happens to everybody. We try to actually help a lot the Global Sepsis Alliance that is based in worldwide, in UK, in the States, and they're trying to make people aware of the what is sepsis, what's going on, what you need to do, what are the protocols. They're doing an incredible job, and we try to be as close as we can with them to make sure that we target this. It's a terrible disease. I mean,
Yeah,
mm-hmm.
it is terrible. No, I used to work in the hospitals with that disease. With bloodstream infections would turn into septic patients, and it's a
And the real question, like when as a biologist, I think like sepsis start from an infection, yes, but not all infections induce sepsis. So what's going on in the body? Why this person or why this bacteria in this body induce sepsis and not in the other one? So that's again my detective molecular, right? So NIVD will interrogate this. NIVD is prepared to say like, what's going on with your immune system? Are you okay? Are you fine? Are you able to? Because like for instance, when you have a disease, you're not sure if you're a candidate for a therapy or not. Okay, so we want to know: is your immune system ready for this therapy? Is not. So we can help a lot on this by asking the right questions. This is the most difficult part. Focus on one disease and what are the most important questions that you want to answer. And then you create the sequence of sensors to answer these questions, and you develop that. It's super cool. I told you, it's
super cool.
It's very, and it's working. So imagine. Yeah. So that's why.
Yeah, it's cool. It's even cooler. It would be
Yeah,
cool
but
doing it, but then it
sometimes there
works.
are new things happening because the innovation is like that. You touch one little thing and something completely different happens, and you're like, okay, now what? But it could be a failure or it could be something new. Working with nano and working with innovation, you never know. So you have to be open-minded. And that's why my team, my scientific team is absolutely amazing. They are like, Sonia, this is not working. It's like, okay, don't worry. We try tomorrow. Let's see. Because we know that we may be facing something new or something that has broken. You never know. Innovation is just like that.
Wow.
You see it, yeah. You come to see it.
I want to see it. That sounds so fun too. It's just the openness of it. Like this could be something different than we expected. Well, and now it kind of makes more sense to me because I was reading about how you feel like this could be really helpful in like underserved communities and areas around the world where they don't have some of these diagnostics to detect these diseases. And now I'm kind of seeing how if like this would be really applicable.
Our aim, and we face every problem or face this challenge, but the aim of NIVD is to create something simple that does not even require any little machine. I don't want to plug anything to electricity. I don't want any microscope. It has to be simple as possible. Therefore, you can do it everywhere. So I work a lot in different countries all around the world. I'm aware of difficulties that people have in order to reach a hospital. And by the time that you reach, the bacteria grow faster than sometimes going to hospitals, right? And some viruses as well. So I think that the final device, has to be something that doesn't need any source of energy. And the energy comes from the nanotechnology. Yeah.
That's even cooler. So someone could go out in a rural area out in nowhere and not even need and just be able to do early detection.
Yep.
Like outside of the hospital environment.
Yeah. That's the idea. That's the idea. The hospitals are where we can actually find more the sepsis, right? But as a preventive tool, you can actually use it everywhere. So that's the whole point for us. To do something that is useful for people.
That's so exciting. Do your plan, or are you at that stage yet of where you want to focus to try to go first with this once you get it out of the commercialization?
As we are very early stage, we are starting, okay?
Yeah.
I need to focus, although every single day I try to unfocus, but I have my team and special person in the team that focus me all the time. But we have started on hospitals, sepsis inducing hospitals or during surgery. We need to obviously validate all of this technology. We need to to complement it with existing data. That's probably the most safest way to start, okay? Because we don't know nothing about early diagnostics. So let's try to work in a place where the cases of sepsis is higher, so you can validate your technology against current technologies as soon as possible. But then this has to be everywhere. We want to bring it to the GPs. We want to bring to rural places. We want to bring it to sports centers because you need to make sure everywhere where you think about it. And sepsis is one of the diseases, but we're working on aside on as well on oncology, but as well autoimmune diseases. People are starting to aware that autoimmune diseases are more and more like diabetes type 1, for instance. Diabetes type 1 is increasing, and there is a reason why it's increasing, okay? And you don't need to be diabetic if we can detect it earlier, right? So you don't need to destroy your pancreas to have the symptoms of diabetes. If we can detect early, there are therapies for this. So we strongly believe that an early detection, okay, by interrogating the system earlier, okay, by catching the first molecules there, we can actually stop the development of the disease. But that will be a dream.
That's so exciting. Now I get it. Now I see why you won that award recently.
Well, yeah, no, this year has been really good. I mean, the team got the Nanotechnology of the Year, International Technology of the Year, the Flux Award, and then 2 awards of as an entrepreneur as well.
Oh, that's exciting. Flux was that I saw that posted. Was that
Yeah,
that was like a thing in Hong Kong? Is that?
yeah. The venue was in Hong Kong, and the prize is let me say it correctly, is the expert in nanotechnology of the year, internationally of the year. So
Wow.
I couldn't go to Hong Kong, unfortunately. I mean, building a company is crazy. And there was a big ceremony to give you the prize, but they sent him to me. I couldn't go there, but I will go next time for sure. Yeah.
Yeah, next time. Yeah. Well, and on that note, so what if you could dream like in 5 years time, like what would NIVD be up to, or where would you all be?
I want NIVD to be useful. Imagine that I go to the GP for checking something and suddenly, oh, let's do this test. And I see my NIVD test. I mean, that would be really fun. But I mean, that's one of the goals. But I want to see this company to be really moving early diagnostics. I want to be creating a platform. The data is important. The data that we are obtaining is super important because it's unknown, the diseases that we have. So I want the early diagnostic be something that is useful. And obviously NIBD on top of it. We want to make it bigger. I will enjoy it as we can. So that's where I would like to everywhere I want to have an NIBD test. Yeah.
Wow.
Absolutely.
No, that's exciting. That's just a small goal. No big
That's
deal.
10 years, 5 years, a little bit too short.
It's just a small goal, completely changing how we diagnose, early diagnostics. That's really exciting. Getting preventative medicine, not
I would love
reactive.
to. I mean, we have to try it. I think that doctors are doing their best with the
Yeah.
tools they have. And when you talk to them and you see the response, it's like, okay, Sonia, just come here, please. It's always happening like this. As soon as you talk, it's like, whoa, come. So we help them already. We are starting going to different places and start helping them. And obviously, our technology is not yet validated. I mean, approved, but it's still early, right? The best way to do is just go to the field and try it, see how it works. And when people see how simple it is that, okay, well, like my mother told me, my mother come to the lab, did the test because everybody can do it. So I said, Mom, do the test. She did it and said like, oh, well, that was very simple. Why nobody else invented it before, right? And you think like that. And it's like, Mom, it took ages to get here.
Yeah. Well, then you need to go in the lab and see.
Are like that,
This is simple.
My mom like,
Yeah. Not.
like why is not being, but there is a lot of knowledge that a lot of people have
Yeah.
bring here, right? So it's, yep.
And
We'll
on
see.
that note, I guess since it is just kind of nano being new, I mean, any like pathways that you foresee, the challenges like FDA or regulatory, is that like are you forging a completely new
Yep.
like
Yeah. Well, FDA is very good. I mean, they have a very good path, so you can actually know which path you need to take. I mean, is all new, but we're using already material that has been FDA approved. Okay, so
okay.
yeah. I mean, I try to make my life simple. So
Yeah.
everything I use is well known that there has no toxicity. That was my previous study, so I know that this material is not toxic because of my own research and from other people's research. Right? We know that it's going to be facing difficulties, and we're going to face what innovation is. I mean, can you imagine the people inventing internet or any other things that change your life, right? It's not easy, but we don't need to stop not trying it, right? If you have the right team, for me, it's the most important, having the right people around you. Yeah.
Yeah. And on that note, I'm just curious with all the data that you're collecting, the things out there, anything that you're finding, I mean, AI specific, are you utilizing that within your process, or is that being helpful for you all?
I mean, we are working on this. I think AI will be a very fantastic tool. I mean, one of the ideas, obviously, the idea is using AI in different moments of when you obtain the data, right?
Yeah.
And I mean, just now we were discussing about this, okay? And obviously, the new data is the powerful of NIVD as well, the data that we will obtain, because it's completely unknown. AI, for me, it's really important at this moment in medicine, and we should all work together because we need to correlate different data from doctors. Imagine you have data from a traumatologist, cardiologist. We need to correlate all of this because there is a correlation between diseases. Nothing happens to you without a reason. So things do not appear in your body without a biological reason. So we can actually help with AI, okay, with our data to actually start helping to link different diseases, right? So
Yeah.
it's an viral infection, actually the source of Alzheimer or whatever, things that at the moment people are not linking. I think that NIVD data combination with AI that we are working right now, it will be a powerful tool. Yeah.
Yeah, that's exciting. That's
Yeah,
where
it
I
is.
see it, being able to pull these
Well,
these things.
I
Yeah.
mean, for me, it's too much. I mean, I can correlate, but it's like, okay, human intelligence is overrated. I need artificial intelligence now. I can't put all this, as well, because the interesting part of AI that is currently being developed is unknown data, right? So AI
Yeah.
with data that is known, it's just training the AI, and AI make links that maybe the human brain cannot link. But what about with unknown data? So we are putting as well the limits of AI. So we'll see where we.
Yeah. We'll see. The future is interesting. Yeah, I'm super excited about to
Yep.
follow you all and where you go. I really appreciate your time today, Dr. Giras. It was really wonderful to learn more about what you're all doing there, and excited to see where it goes, to see these out there in the world.
Soon,
And
soon,
thank
soon.
you for the work that you're doing and just the
Thank
passion
you.
that you put behind it. Thank you for taking it out of the lab, you know, and
Yeah.
putting it out in the world. And it's not easy. So I really appreciate that. Yeah.
Well, thank you so much
And
for your time and this opportunity to talk to you. It's been fun.
yeah, and then where can people find you? What's the easiest way to just keep track of tabs of what you're all
Oh,
doing there at
we are
NIVD?
we are doing what we have a website. It's www.nivd.world. The social media as well. I mean, we are people helping us on this to reach, because it's the way that we communicate right now. You should come to see the test.
If I've only been to Spain once, but I was in Madrid. But if I'm in Barcelona, there's no way I'm not coming in there and seeing
That's the
what is going on in this lab. Yeah, super curious. Thank you so much. Have a great rest of your day. Appreciate your time.
Thank
All
you
right.
so much for the opportunity.
You've been listening to Stories in Life Sciences, sponsored by MedAccess AI, where we explore people, ideas, and breakthroughs shaping the future of biotech and healthcare innovation. To learn more, visit medaccessai.org and subscribe on YouTube to watch the full conversations and new episodes as they launch. I'm Christopher Wilson. Thanks for joining us. Until next time, keep shaping the future one story at a time.